plant growth regulators, we are telling that there are 4 important group of chemicals. That is your auxin, cytokinin, gibberellins, and abscisic acid. So these are our growth regulators. And why is these growth regulators necessary during our tissue culture is only the growth regulators can bring about the differentiation and organogenesis of the tissue. If my tissues have to grow or if my cell has to grow, it can be brought about only by the growth regulators. That is why we add 2 or 3 growth regulators which are necessary in the media. And also remember, the ratio of hormones required for root or shoot induction varies considerably. So this is why we require the plant growth regulators. We shall define what are plant growth regulators. Plant growth regulators are compounds which are required in low concentration. Okay. These are required in low concentration and they are capable of modifying the plant growth. Okay, either growth or even the morphogenesis of the plant. Morphogenesis. So what do you mean by morphogenesis? It is the development of the tissues or organs into a particular shape. That is called as the morphogenesis. So my plant growth regulators are required in small quantity. Though they are required in small quantity, they are very essential because they are important for the growth and morphogenesis of my plant. So usually into a media, we require at least 2 to 3 growth regulators we add. And also remember, the ratio what we add is also important. What is the ratio of the growth regulators we are adding? That also is an important. So just a small differentiation. We interchange plant hormone, plant growth regulators. Even throughout my class also I'll be using these two, interchanging and using. But actually plant hormones are naturally occurring in the plant. Plant growth regulators means they are both naturally occurring and synthetic. That is when we do it, we start synthesizing in the laboratory. That is called as synthetic, and that actually refers to the plant growth regulators, but usually we keep interchanging it. All plant hormones are growth regulators. So all your plant hormones can be your growth regulators, but not all growth regulators are your plant hormones. Why? Because I can do different type, I can synthesize a different variety of compound, which is called as growth regulators, which may not be available naturally. So remember, all plant hormones are growth regulators, but all growth regulators are not plant hormones. So in my class also, I'll just keep interchanging these words. Now we shall go to our first growth regulator, that is auxins. So we have already studied that Wendt isolated auxin from the tips of the coleopetals of the oat leaves. Coleopetals means the tip of the region. If this is the plant, okay, this region, if this is a plant, the tip of this region, okay, it is called as the coleopetals. So from there it was isolated. So where are these auxins produced? Auxins are the natural hormones of the plant. So where are they produced? They are produced in the apex of the stem, that is in the tip of the stem, and even in the roots also they are produced. So from there, wherever they are produced, okay, in the tip of your roots or in the apexes, wherever they are produced, they migrate to the region wherever they are required. So they are produced in and oxons, no, it is not soluble in water. You have to dissolve it in ethanol or dilute NaOH. So what is the main function of our oxen? The main function of our oxen is elongation. And we also came across a word known as indole acetic acid. This is one of the common oxens which are used. So as we told that it is present in the shoot tips and even in the root tips, and they are transported to all other region. So if you look at the function, or let me go into this. If we look at the types of oxens, there are natural oxens and also there are synthetic oxens. Natural means they are present naturally in the plant. Synthetic oxens means it is man-made oxens. So the most common is the indole acetic acid, indole butyric acid. So they are isolated from the plants because they are present naturally. But synthetic is what man has created. That is, you have naphthalene acetic acid and 2,4-D. This 2,4-D is very important. That is, it is 2,4-dichlorophenoxyacetic acid, which has been asked for 2 marks question. So these are what they are. They can be used as herbicide. 2,4-D is used to kill dicot weeds. Weeds means we don't require that plant, unnecessary growth of certain plants. These are called as weeds. So weeds. So we can use this 2,4-D, which is a synthetic form of oxen, to kill the dicot weeds, but it is not effective against the monocot plant. Most of our monocot plants are what are crop plants. So the function is, see, that it establishes the apical basal polarity of the seed embryo. We know that whenever there is a seed, it has a polarity to develop into a shoot, and then it is bipolar to develop into root. Induces vascular tissue differentiation. It mediates phototrophism, very important. Promotes the formation of adventitious roots. This is also important. Stimulates fruit development, and it is used for cloning in plant tissue culture. So remember, whenever we are growing a plant in a tissue culture, okay, remember this is your explant and you are growing the plant in the some media. So what happens if I want this plants to produce the root? I have to use what? I have to use the auxins. Okay, so auxins are used for the production of the root for your tissue culture plant. And also it stimulates the fruit development. We can see here some of the important functions of the auxins. A common feature of auxin is the property, even it can induce cell division also. So in nature, hormones of this group are involved in activity like elongation of the stem. It elongates the internodes. It brings about phototrophism, as we have mentioned here, apical dominancy, and then it brings about the rooting in the plant. So these are the important functions. Here you can see that when the auxins were added, this type of adventitious roots have been produced in the plant. Similarly, we can see that when auxins were added to strawberry, the strawberry size increased, and it has phototrophism. Phototrophism means, you know, that it is towards attraction towards the light. So here what happens, see, this is a apical region of your plant. Okay, if this is the plant, this region, what happens whenever the light is there, it bends towards that light. That is what we have drawn it like this. So what is there in these apical regions? As we told, in the apical regions we have the indole acetic acid. Indole acetic acid is nothing but your oxen. Okay, so now when there is light, no, this all are cells. Okay, all the cells will be having the indole acetic acid. When the light falls on this apical region, all the indole acetic acid, they'll move to one direction, all in the opposite direction of the light. So your less amount of indole acetic acid there, all have got region of the light. Why? Because what is happening here, more auxins are there and it inducing the growth in this region. So there is severe multiplication of your cells in this region. So automatically what happens, the other region which is having less auxin will bend. This is the phenomena, phototrophism. So this is the normal growth which has been here, but there is excessive growth occurring. That is why your plant bends like this towards the light. And also like apical dominance we mentioned. So it is not clearly visible. Okay, but this is the apical bud. Right? This is your lateral buds. Here what you're seeing, no, this is all your lateral buds and this is your apical buds. Now scientists did an experiment. What they did here, you see here, the apical bud is removed and it is replaced with a gelatin block containing indole acetic acid. They removed this apical bud and on this what they placed an gelatin. What is this gelatin containing? The gelatin is containing indole acetic acid. So what happened? They observed that only this apical region started growing. The plant started growing like this. There was no lateral growth. No lateral growth. It is all absent. Lateral growths are absent. In the next experiment, what did they do is the apical bud was removed and replaced with again gelatin block. Okay, I placed the gelatin block. Now this time, the block is not having any indole acetic acid. Now what happened? The plant did not grow like this straight. It did not grow. Instead of that, you see here, lateral buds started developing. So if I want a bushy tree or plant, so I can cut off this region so that always your lateral stems can grow. So this is known as the apical dominance of the importance of the auxins. So remember, the main important point is it is inducing the root formation in your tissue culture plant. That is what you have to remember, most important point. Apart from these, whatever I mentioned, see, it promotes flowering. Actually, I'll tell you about this later on. There is a story about this. We'll run short of time. Next, coming to gibberellins. These gibberellins are also known as gibberellic acid, or simply they are mentioned as GA, and they are also produced in your apical buds, in the roots, young leaf and seeds of your embryo. Now this gibberellin have shown to stimulate blotting, which is a rapid stem elongation and flower initiation. I'll explain to you what this stem elongation and flower initiation, blotting all means. So how was this gibberellin identified first? It goes back to way back in something 1900s. What happened was in Japan, in rice plants, a disease called as Bacne disease or foolish seedling caused by a fungus, Gibberella fujicurea, resulted in plants that grew very tall. So now what happened? I have my paddy plant, okay, rice plant. Now it is growing, but along in the soil, what is that? There is a fungus, and this fungus, it is inducing a chemical, okay, towards this plant. And this rice plant, what happened? It started growing very tall. And all the paddies were here. Now what happened? It is, you know how the paddy plants are. Okay, so lean they are. And now what happens? This started drooping because of the weight of the paddy grains. And whenever there was breeze or heavy wind, what happened? Because they are very slender, they started to fall off. Now they were wondering what is happening. Then they came to a conclusion, this is because of this fungus, okay? This fungus Gibberella fujicoria, which is actually producing the chemical which is being passed on to your plant, and that plant is increasing in the size. So later on, here you can see gibberellic acid made by the fungus caused the rice plant to overgrow. Then later on, it was found that plants also make gibberellic acid, and that apply it to the plants caused growth. So what it means is, okay, they found that this fungus is also producing at sun. But also later on they came to know that our plants also, by naturally, they are producing this gibberellic acid. So there are at least more than 100 of this gibberellic acids which are present from the fungi and higher plants, which it is known so. You can see here this plant, it is very dwarf. It is a bonsai. That is, here what has happened, they have removed the GA. So it is short, this plant, because it is having a very reduced amount of gibberellin, and that is why it is a short plant. It is a bonsai plant. So I can treat even my grapes. This is my normal grapes. But what the scientists have done, they've treated this normal grapes with the gibberellin, and they saw that these grapes became large in size and they elongated. And most of you have seen such type of grapes, which are long grapes and elongated in size. So what happened was, like, it is agricultural boon to them to get such a big grapes. And one more you have to remember, in gibberellins, there are no synthetic forms. Like in auxin, we told, no, man-made ones, artificial ones, auxins, like naphthalene, acetic acid, 2,4-D and all. But here such type, no synthetic form of this hormone is present. And of all the hormones, okay, of all the gibberellin hormone, GA3 is the most commonly used in the plant tissue culture. So what is its function? Stem growth and elongation, then blotting and flowering. You can see here, once again, you can see here, this is the untreated mutant plant maintained as a dwarf. This is a very small plant, that is what it's called as dwarf. But see this plant, 22 days after being sprayed with dilute gibberellin solution, this plant reached the size of, now it is quite tall. It is no more dwarf. That is a plant which was dwarf. What did the scientists do? They added the gibberellin to this, and then 22 days after being sprayed, they saw that this dwarfism was lost and the plant reached the normal height. And it is also induced in seed germination, dormancy, sex expression, fruit growth, parthenocarpy. These are all the important functions of the gibberellins. And one more functions, you can see that this plant, no, it is rosette, this shape. So when you induced gibberellin, what happened? It started growing, well spread, tall, it started flowering. And this is known as blotting, what I mentioned here. Okay, blotting and flowering. If your plant is in the rosette form, it's not flowering, you induce gibberellin, that is your GA3, which is commonly used. Okay, you get this beautiful plant. which is grown tall and it just also started flowering. So you can, with these two hormones itself, you can come to know that how important the hormone is. Without this hormone, there is the plant development, the organogenesis will not occur at all. And we saw that the father of tissue culture, Hamberland's experiments, how it failed and how he told that there is some enzymes which is required for the growth of the plant. And today we know that what are these, what he meant by enzyme. He meant it is a hormone or the growth regulators. Coming to cytokinins, very easily you can remember cytokinins. Cyto means cell, kinins means division. So cell division, the main function of your cytokinin is the cell division. And the cytokinin was first discovered as kinetin by Shoup and his co-workers, and this naturally occurs in the plant. So this cytokinin is an adenine derivative which are mainly concerned with the cell division. They are all derived from the adenine. One second. A-D-E-N-I, adenine. So your cytokinin is derived from this adenine and its main function is cell division. And it can also bring about modification in the apical dominancy and shoot differentiation. So you have to remember this ratio whenever actually this auxin is always used for your root formation. Okay, this part you have to remember. And your cytokinin is always for your shoot formation. So there are again different type of cytokinins. Okay, like this most commonly what is used is DAP, 6-benzyl aminopurin, and even the cytokinin are not soluble in water, they are dissolved in HCl or NaH. So the cytokinin in plants promote the cell division by stimulating the production of proteins needed for mitosis. And also they induce the callus formation in your plant tissue culture. So some of the commonly naturally available cytokinin is the zeaxanthin. It is a natural substance with cytokinin activity. There are some synthetic compounds with cell division promoting activity. With synthetic means it is man-made. And natural cytokinins are synthesized in regions of rapid cell division, okay, wherein the root, apices, shoot buds, young roots, and etc. Cytokinins are produced in actively growing tissues. That is what here also, your roots, embryos, and fruits. Cytokinins work together with auxin to control the cell division and differentiation. That is what I mentioned in your previous slide. Cytokinins slow the aging of some plants by inhibiting the protein, stimulating RNA and protein synthesizing, and mobilizing the nutrients from the surrounding tissue. These are some of the important function of the cytokinins. But the first point what you have to remember is that it is used for cell division and it is also used for callus development. This point you have to remember, girls. Once see this one. The ratio of auxins and cytokinins is always important with respect to the morphogenesis of the plant. Whenever there is the cells and tissues, when they are undergoing division and they're forming a particular shape of your plants, that I told you it is morphogenesis. So for a morphogenesis of a plant, it becomes very important who your auxins and your cytokinin. And also, as I told you, your cytokinins, it is involved for embryogenesis, callus initiation, root initiation, that is more important, and shoot proliferation. So these all points you have to remember for your cytokinins. The last one what we are going to see is the abscisic acid. Now this abscisic acid, it is also called as ABA. When it is present in the culture media, it has dual function. It can stimulate or inhibit the callus. So you have to choose whether this ABA is useful for the development of the plant or not. So based on this, again, trial and error method only, you can use this abscisic acid. Particularly, it is an inhibitor of plant tissue culture, but in some plants they have seen that it has the positive effect also. Another important function of this abscisic acid is the major effect of abscisic acid is its capability to signal that it is undergoing water stress. Like whenever the plant is growing in some drought condition, it can send a signal to the plant. This hormone can send a signal to the plant that we are under water stress. No water is available, so don't lose your water. So when a plant is undergoing water stress, the ABA levels are increased. They increase. So the water level has come down. Okay. Thereby what happened? Your ABA level has increased. So what does this indicates? The ABA levels are increased dramatically, leading to the stomatal closure, thereby preventing the water loss. So what it does, it increases in concentration, and you know what are stomatas, the guard cells and all, it closes the stomata, so that from the stomata you don't lose any water. This is an important function. But from tissue culture point of view, it is hardly has much function to do. Next is again ethylene. Ethylene is also not that very commonly used. Even these two, this abscisic acid and ethylene, from plant tissue culture point of view, they don't have much functions to perform. Otherwise, in a general growing plant and outside, you can see that they have lot of function, like it is useful for water stress. Ethylene is a gas, and you all know that it causes the ripening of the fruit. For example, if this is an unripe fruit, ethylene, it induces the ripening of the fruit. Similarly, the starch in presence of amylase is converted to sugar, and the chlorophyll, the green color chlorophyll, because of the enzyme hydrolase, it is converted to anthocyanin. So these are the some of the example which ethylene and abscisic acid have in a normal plant. But when it comes to tissue culture level, hardly they have any function. So when we come to our growth regulators or growth hormones, your take home message is auxin. The most important function of your auxin is the development of root. The most important function of your cytokinin is the shoot. That is what I told. The ratio of auxin to cytokines. If the ratio of your auxin is more and your cytokine is less, what happens? It induces the root formation. On the other hand, if your cytokinin ratio is more when it compared to your auxin, okay, it induces shoot formation. This one you just have to imprint in your mind, never to forget. Okay. Gibberellins, it is involved in cell enlargement or cell elongation also. Abscisic acid, it depends. Okay. It depends whether it is a stimulator or inhibit, or it can inhibit the callus growth. Dependingly, you can have to use the abscisic acid. Otherwise, it indicates the plant stress hormone. Ethylene we don't use because it is only used for the fruit ripening. So these are the important hormones which are naturally produced by the plant, and we have to use them in different concentration or the quantity and quality, so that we induce it into the media for the development of the plant, for the cell division of the plant, or for the formation of the root, shoot, cell enlargement, cell progression. So all this is done by our hormones. So before we could wind up, we will see into one more substance which is very important. So you were studying about inorganic nutrient, macronutrient, micronutrient, organic natural materials like your coconut water, potato. You saw the importance of charcoal, antibiotics, vitamins importance, amino acids importance. You saw now the what are the hormones which are important for my plant. So now we shall look into the another system called as the solidifying agent. So you know, once I tell what is the solidifying agent, what you use in your tissue culture or even in your microbiology, you immediately what strikes to your mind is the agar, because you have used it. So this solidifying agents, these are gelling agents. Okay, it is called as gelling agents or solidifying agents, and it is commonly used to prepare semi-solid or solid tissue culture media. Now why we are using is, suppose say now if this is my tissue culture vessel and I'm growing my plant here, and what happens if this is filled with only the liquid? What happens to my cell? It gets immersed in this liquid. So once it gets immersed in the your liquid, what happens ultimately? There is lack of oxygen for this cell to perform its function. So it dies in the liquid media. So what we are doing, we are using the solidifying agent or gelling agent. So what happens? There is a thick solid form which contains all your nutrients, and your plants can be kept here so that it will not immerse into it. So there is an air exchange, and then it will not die. If it is a liquid media, it will get submerged into the liquid. Ultimately, there is no oxygen and your plant can die. So the most commonly what we use is the agar, 0.5 to 1% agar. This agar is a polysaccharide which is obtained from the seaweed, and it is inert. That is, it is not going to react with any of your media. Its main function is to just solidify your media. That is why agar is most commonly used. It is very inert. It is not going to interfere with your media at all, or not the growth of your plant. And also they are not digested by plant enzyme. That is, I told you, these plants, no, they will be secreting certain residues. I told you, like phenolics. When the phenolics are secreted, what happens? Your plant, this media turns black in color. These are the impurities, waste, which can be removed. One method of removing these impurities is you can use activated charcoal, or you can go for subculturing. I'll tell you this all in detail when we talk about that chapter. So my plant, what it is doing, or my tissue culture cell, what it is doing, it will be secreting certain enzymes. So this agar has no effect on this enzyme. You produce the enzyme, but still I'm strong. I will be like a solidifying agent. That is what is the function of the agar. Now suppose if you want to study the nutritional content of your tissue culture, that time you cannot use this agar, because this agar will have some impurities, okay, from like calcium, magnesium, sodium impurities it has. That time what you have to do is you have to remove these impurities, and then you have to do the nutritional studies, which is not, it is out of scope of our syllabus. Okay, but just remember, it can be, the impurities can be removed by using the double distilled water. You rinse it for 4 hours in ethanol and dry it at 60 degrees centigrade for 4 hours. So all the impurities will be removed, and you can study the nutritional content also. Apart from agar, you can use another chemical agent or the solidifying agent, which is gelatin, which is used at a concentration of 10%, but this is limited use because it starts melting at 25 degree. So at 25 degree itself, if it starts melting, what happens? Is it a gelling agent or is it a solidifying agent? No. So it has a limited use. Apart from your gelatin, we can use agarose, we can use alginate, and we can use phytogel, phyta-agar. So these are some of the solidifying agent or gelling agent or supporting system, so that it acts as a support for your cell or tissue or callus to sit on the media, which is containing all the nutrients what we spoke about. And here the last one, you can see mechanical filter paper bridges, wigs and wraps. Sometimes what happens is, this is particularly used in when we are using this liquid, when your media is in the liquid form. So when it is in the liquid form, imagine this is all the liquid form. Now I have to place my tissue culture plant. Now if I place, what will happen? It is just going to get immersed into it. Instead of that, what I'll do is I'll make a filter bridge in filter paper or wigs. In chromatography, you know that how we made the wigs. So we can